Okay, now today we talked last time about glycolysis, and I think we also got to oxidative phosphorylation, right? And so if we have a molecule of glucose undergoing complete oxidation all the way to carbon dioxide and water using glycolysis and the Krebs cycle and the oxidative phosphorylation process, how many ATPs of energy were net from that? 36, right? Okay, and that assumes, of course, that oxygen is present, because without it, what happens with oxidative phosphorylation? It stops. And if it stops, what else stops? The Krebs cycle, right? Because the Krebs cycle relies on oxidative phosphorylation to oxidize NADH and FADH in order to keep the Krebs cycle operating. Okay? All right, so there we go. Now, what if we take away the oxygen? How much ATP is net for glucose in that case? Just 2. And where is it coming from? What pathway is producing the net gain of 2 ATPs for glucose in the absence of oxygen? Glycolysis, right. Now, glycolysis, as we said, can operate anaerobically in certain types of cells. What were the types of cells that we mentioned? Liver cells, skeletal muscle cells, smooth muscle cells. All right. Now, how do they continue to operate? Well, in glycolysis, we also have some NADH produced. And of course, if you have no way to oxidize that back to NAD, well, that process would also shut down. So how do we keep it going when we're under anaerobic conditions? Well, this process under anaerobic conditions is going to be accomplished by fermentation. All right, so fermentation is able to oxidize NADH back to NAD in those cells that can undergo anaerobic utilization of glucose. All right? And fermentation typically operates under anaerobic conditions. Now, what does it do? Well, in addition to oxidizing NADH back to NAD, it also takes the pyruvate, and where do we get the pyruvate? Where did that come from? That was the end product of what process? Glycolysis, right? Because where did glycolysis start? You fed in a molecule of glucose, what came out the other end? Well, and 2 molecules of pyruvate, right? Well, what it does, as it's oxidizing NADH back to NAD, it's converting that pyruvate into what we could call a fermentation product, and one of the most common ones, lactic acid. In muscle in particular, that's what you're going to get. All right? So when muscle is under anaerobic conditions, pyruvate is converted into lactic acid as NADH is oxidized to NAD. And we have to have NAD in order to keep that glycolysis operating. If you run out of NAD, glycolysis would shut down as well. Okay? Now, of course, what's the problem with, and we'll talk more about this when we get to muscle contraction, what's the problem with accumulating lactic acid in muscle? Well, have you ever used muscles that you haven't used for a long time that didn't disuse, and how do you feel a day or 2 later? Sore. Well, there you go. That's due to that accumulation of lactic acid.
But can't make you really sick also.
Huh?
You can't build up with like lactic acid in your muscles, make you really sick.
Well, yeah, it can, but the thing of it is you're only going to go so far, because when the muscles run out of energy, they become fatigued, they don't contract, and you got to wait for the lactic acid to be taken out. It goes to the liver and it gets reprocessed back to glucose. But still, it takes some time. Now, theoretically, it's 30 minutes. Well, you know, if their muscles have been in issues for quite some time, it takes longer. But anyway, there you go. All right, so under anaerobic conditions then, in order to keep glycolysis going, we have fermentation, which oxidizes NADH back to NAD and converts pyruvic acid or pyruvate into lactic acid. All right? Now, simple as can be. All right, well, let's take a look at this next slide, which kind of puts it all together, in a sense, when it comes to getting energy. And we're looking at kind of an interconvertibility, if you will, of various types of metabolism. And I know this probably isn't easy to see, so we'll just leave it there for a while, and then we'll write on the board. All right, now basically what this is trying to show us is that we get energy, or we can get energy, not only from carbohydrates like glucose, but we can also get energy from amino acids, from the breakdown of proteins, and we can also get energy from the breakdown of fats. All right? And typically what cells are going to do, they're going to go with the course of least resistance, which means glucose is what they're going to use first. But if, for example, you're in starvation, then what are you likely to use next if you run low on glucose and the cells need to keep producing energy? They're going to use fats. All right? And then as a last resort, they'll go to proteins and start breaking them down into amino acids and using those. All right? And by the same token, if you've got excess carbohydrate, you can take the excess carbohydrate and convert it into fats and store it. Or, in some cases, we can actually siphon off materials out of the Krebs cycle and make amino acids. So there's always that possibility as well. But cells are going to go with glucose first. It's the easiest thing to use to make energy. But let's just say a word or two about how we get energy from fats and how we get energy from amino acids. So if we just, come on. There we go. All right, now let's take the old fats, shall we? Now, what are we primarily talking about? Well, we're primarily talking about neutral fats or triglycerides. Now, what is a triglyceride made of?
Two fatty acids and three fatty acids.
Yeah, and what else? Well, first thing it's going to get broken down into glycerol and 3 fatty acids. That's what a triglyceride is, okay? It's a glycerol and 3 fatty acids. Now, the glycerol can go directly into glycolysis. So you can fit that one into glycolysis, and you can convert that into pyruvate. And then where does pyruvate typically go once you've got it in glycolysis? Into the Krebs cycle. All right, and of course, when that chugs around, out comes, of course, carbon dioxide and water and some energy in the form of ATP. All right, now, what about those fatty acids? Well, fatty acids first undergo something called beta oxidation. Now, what that does is essentially break off 2 carbons at a time. All right? So let's say you had a 16-carbon fatty acid. You can break off 8 2-carbon fragments, right? Okay. Now what are we going to do with those 2 carbon fragments? Well, they are going to get converted into acetyl-CoA. Now, where have you heard of acetyl-CoA before? Into the Krebs cycle we go. Remember, acetyl-CoA was what pyruvate went into the mitochondrion, got converted into acetyl-CoA, and that plugged into the Krebs cycle, right? Remember that? Yes. Well, you take those 2 carbon fragments from your fatty acids, go into the mitochondria, acetyl-CoA, plug it in the Krebs cycle, away you go. All right? Well, it's simple. And as you can see, you can get potentially a lot of energy there. Because if you could have, let's say, 24 acetyl-CoAs coming out of one triglyceride, that's a number of trips around the Krebs cycle, right? That's 24 trips around because every acetyl-CoA gives you 1 turn of the Krebs cycle. And that produces quite a lot of energy. Okay? Well, that's simple. Potentially there's a lot of energy that can be had from this. But there's also a downside to it. Because if you start really breaking down lots and lots of triglyceride, one of the things that you can end up forming are some nasty things called ketone bodies. And those can lead to, if you get too many of these, you can end up with ketoacidosis. Somebody drilling away or something. All that noise. All right, with ketoacidosis, which, you know, can actually be fatal if you get too much of this stuff. Makes your blood too acidic, and, you know, and there you go. Now, when are you likely to get into this kind of a thing? Well, in extreme starvation, where the individual's really mobilizing lots of fats, or in people like with uncontrolled diabetes mellitus, they're more susceptible to ketoacidosis because they're breaking down fats. Lots of fats. Well, you know, they got to get energy somewhere, and that's one way to get it. Okay? Now, there you go. Now, not all cells can really do all this, but nonetheless, a number of them can. And so it is a way of getting some energy. Now, if you run out of fat, what are you going to go to next? Proteins. All right. Now, when you start breaking down proteins, you do start wasting away because typically structural proteins, like proteins coming out of muscles and other organs, are going to be the source. So if we consider the protein business, all right, first of all, of course, proteins have to be broken down into amino acids. And then it's fairly simple because a lot of these amino acids can plug right into the Krebs cycle and undergo oxidation to carbon dioxide and water and produce energy. All right? Because remember now, as we said, that there are some of the Krebs cycle intermediates like alpha-ketoglutarate, alpha-ketoglutarate is one of those intermediates in the Krebs cycle. You can actually siphon that off and use that as a starting material to make amino acids. So if you've got excess carbohydrate, then, you know, you can get it down to the alpha-ketoglutarate, siphon some of that off, and then, you know, start making amino acids that way. So you can convert, you know, you can actually convert sugars into amino acids. Or vice versa. And of course in starvation, this is what's likely to happen, is that proteins get broken down to amino acids then get plugged into the Krebs cycle and oxidized for energy. Okay? You know, the same deal like with fats. I mean, you know, too much sugar, you can convert that into triglyceride as well. You know, kind of reversing what we had up there a bit ago. So there you go. All right, now it's simple enough, isn't it? So what are cells going to use first for energy? What's their primary choice? Glucose, right. And if they don't have that, they'll go to fats. And if they run out of fats, they'll go last to proteins. All right? So there's that pecking order. And of course, what happens? Well, you have people with uncontrolled diabetes. Well, you know, they're probably going to deplete their fats, and then they're going to break down proteins. And then you know, eventually they're going to have nothing more, and, you know, and there you go. But we would hope we would get to that. Anorexics, you know, they can get that way too. Also, you know, people who are, you know, in extreme starvation situations, and of course, examples of that today in parts of the world. So, there we go. All right. So, of course, there's just one thing. I mean, you know, some cells, like red blood cells and neurons, well, they have to have glucose. And so if you can't get glucose, well, you're just kind of in bad shape. Well, enough for carbohydrates, lipids, and proteins. Let's go back and take a look now at nucleic acids. And so, let's just chat a little bit about DNA. And then we'll have some fun with that. All right, so DNA. Now, remember what the letters DNA stand for. What is it? Deoxyribonucleic acid. That's right. All right, and get lined up with that. Now, remember that DNA is made by stringing together what we call nucleotides. And those nucleotides are composed of the sugar, and in the case of DNA, it's deoxyribose, which is one of the pentoses, remember. And they also have a phosphate attached to it, and that makes up what we commonly think of as the sugar-phosphate backbone. And then we have attached to the deoxyribose and the phosphate some base. And there are 4 different bases, 2 of which are what we call purines and 2 of which are pyrimidines. Okay? All right. So, here we have the bases and the way they pair up in the molecule. We have adenine and guanine, our purines, if you will. And then we have thymine and cytosine, which are the pyrimidines. All right. Now, in the molecule, adenine pairs with thymine and cytosine with guanine. All right. So we have those. So a nucleotide, DNA nucleotide, would be like deoxyribose, phosphate, and adenine. Deoxyribose, phosphate, and thymine, and so on down the line. Just 4 really nucleotides. That's all there are. And that's enough when you string them together in a huge, almost infinite combination. You can come up with the parts that we need. And so here's kind of a quick representation of a nucleotide with the sugar, phosphate, and base. Now, when we string them together, we end up, in the case of DNA, with what's called a double helix. In other words, 2 of these chains, one facing the other. And so here is kind of a representation of that. So here would be one of these sugar-phosphate chains with their bases. Here's the other. And the dotted lines between the bases from one chain to the other, well, they're one of your favorite things to form the cockles of your hearts. Those are indeed hydrogen bonds. All right? So, there we are. So the hydrogen bonds hold these together into what we call a double helix because it will have this spiral-shaped appearance to it. All right, so there it is. Now that's DNA. Now, a couple of other terms that we need to note, and I think these are probably in your outline, but since I'm operating off a different slide set today because I forgot my thumb drive downstairs, I'm going to have to wing it a bit. So, here we go. Now, we have some terms that we need to throw up here, and we'll come back to this in a moment. And so, let's just write some of this on the board. What have we got? Well, we have the term gene, right? I think you've got that in your outline, don't you? All right, now what is a gene? Well, it's just a segment of the DNA molecule. segment of the DNA molecule that codes for the production of a protein. All right? Segment of the DNA molecule that codes for the production of a protein. Now, DNA is an extremely large molecule. And remember, in the nucleus of cells, we had that chromatin material. What was the chromatin? Strands of DNA wound about, what were those little spindle-shaped proteins called? Histones, right? Remember those? All right, and when the cells got ready to divide, what happened with the chromatin? It condensed into those bodies that we call chromosomes, right? Yes. All right, so chromatin, DNA, and that's a very long molecule, and a segment of that DNA that actually codes for a protein is called a gene. All right? Now, if you string together a whole bunch of genes in a linear sequence, one right after the other, what do you end up forming? You end up forming a chromosome. So a chromosome is really nothing more than a linear sequence of genes, one right after the other. Now, I don't know how many people remember audio and video cassette tapes, right? Remember those? It's not been that long ago that they were quite popular. All right, now, let's take an audio cassette for a minute. Let's say it was a cassette of an album. What did you have? You had a linear sequence of songs, right? One right after the other. Well, a song would be somewhat analogous to a gene, all right? So a gene is just part of that tape that codes for the production of a protein. And so you got one gene, and then there's a little space, and then there's another gene that comes right after it, and another one, and so on down the line. And there can be thousands of genes on a chromosome. All right? There you go. So genes code for proteins, sequences of genes organized into what we call chromosomes, and as well we know how many of those chromosomes did we find in human somatic cells. 46, right. All right. Now, there we go. All right, now, when it comes to actually replicating stuff, and when during the cycle does replication take place? During the S phase of interphase, right? Remember that? That's when the cells replicated the DNA. DNA replication occurs in what we can call a semi-conservative manner. Okay. Now, what does conservative mean? If you're conserving something, what does that mean?
Saving.
Saving it. Semi means what? Partial. All right, so we're saving part of the original as we replicate the DNA. What does that mean? Well, let's put up a bit of DNA here, and then we'll replicate it and just see exactly what that means. All right, so let's just have these two strands, and so we'll have one here, and we'll put some nucleotides on this one, and we'll stick another one over here. I want to leave a little bit of space this time. And we'll put some nucleotides over here. All right. Now, let's start filling it in. Let's say it goes something like A, T, G, C, C, A, T, T, A, C. All right, now that's on one strand. Now, what do those letters represent? They represent the bases: adenine, thymine, cytosine, guanine. Okay, remember those are part of the nucleotides. In fact, so the line over here, the straight line, well, that's the sugar phosphate backbone. And that's the way they're strung together. All right, now, if there's an adenine at that location on that chain, what's going to be over here on this one? Thymine, right? Because remember, adenine and thymine pair together, okay? All right, so we've got a thymine here. What's going to go over here? Adenine, all right. Guanine there. What goes here? Cytosine, all right. Cytosine here gives me a guanine there. And let's see. All right, let's skip the cytosine, so we'll put another guanine there. All right, adenine here gives me a thymine. Thymine here gives me an adenine, thymine, another adenine, adenine, a thymine, and then finally a cytosine there. There's a guanine. There it is. And then between these two bases, kind of like the rungs of a ladder, we'll dot in some blue lines to represent the hydrogen bonds that hold it all together. And then we take all this and we wind it around some histones. We end up with chromatin, or if we condense it down, we can come up with chromosomes. All right, so there you go. Now, replicating it. Now, as we said, it's a semi-conservative process. So what we have here right now are these two original strands, and these originals we can call parenteral strands. All right? So I've got a parenteral strand there, and I've got a parenteral strand there. Those are the originals. That's what we're starting with. All right, now we come to the S phase and we want to replicate it. Now, how is it going to operate? Well, one of the first things that's going to happen when we get ready to replicate this, we're going to have an enzyme come in here and unzip part of the molecule. So what's it going to do? Away with those pesky hydrogen bonds. Because now what we want to do is get these to separate a little bit of space. Okay? Now, what are we going to do? Well, we're going to start synthesizing a new strand that's complementary to each of these. In fact, we're going to need two new strands, and they're going to be based on the pattern that you see right here. So, I don't want to use blue, better switch to green. All right, so let's suppose over here, there's my sugar phosphate backbone, even though it's got a nucleotide at the time. All right, so now what's going to go here? Adenine pairs with what? Thymine, right? All right, so what do we got? There's another adenine, a cytosine, guanine, guanine, thymine, adenine, adenine, thymine, and guanine. All right? Put back the hydrogen bonds. All right, now, before I go any further, what do you got here? Looks like it's a copy of that, doesn't it? Okay? Well, what do you suppose is going to go over here? Whoops, I don't want to do that. Go back to my color. All right, so what am I going to put over here? Well, the complement of what I've got from that parenteral strand. So, starting up here, I'll have an adenine, thymine, guanine, cytosine, cytosine, adenine, thymine, thymine, adenine, and cytosine. All right? That looks exactly like that, doesn't it? All right, so what have I got? I now have two DNA molecules, two double-stranded DNA molecules. Here's one, here's the other. What is the one made out of? It's made out of a parenteral strand and a newly synthesized strand. And the other one is exactly the same way. I have a parenteral strand and I have a newly synthesized strand to go with it. And of course, let's come back, pop in our hydrogen bonds. It's all said and done. All right? Now, it's a little more complicated than that, but not much. So, every time you replicate DNA, what do you end up with? You end up with this double-stranded molecule, half of which is conserved. It's a parenteral strand from the original. Half of which was newly synthesized to be complementary to that original parenteral strand. So you've got two copies, and they're both identical. Are they not? Well, we hope. All right, there you go. Now that's simple, isn't it? Isn't it? Where, I wonder, in the cell does this replication of DNA take place? Where would it be happening? Where do you find the DNA typically in the cell? Huh? Nucleus, right. Okay, there you go. Well, that's simple, isn't it? Now, if this cell we're getting ready then to divide, we've got these replicated DNA, so one copy is going to end up going to one of the daughter cells, and one goes to the other. They each getting the same thing, and they're going to end up being genetically identical. All right, so there you go. All right, simple enough, isn't it? Okay, well, I'm going to get rid of one of these, and I'm going to keep the other one, because I'm going to use it here in a bit. All right, so let's just come back and take a look, because I think our next slide essentially shows us this replication business. And, in fact, it does, because here we have, in the dark blue, I think it is, the original DNA up there at the top of the screen. And as you notice then, where it's replicating, it's been spread apart, and newly synthesized strands in light blue are being produced that are complementary to that original parenteral strands. And so we end up then with two double-stranded molecules, half of which is old, half of which is new. Isn't it nice they use blue? You know, or like something borrowed, something blue, that sort of thing. Well, anyhow, there you go. All right, now that's simple, isn't it? All right, so there's the replication of DNA. Now, what are we going to use DNA for? Well, we're going to use it to bring about protein synthesis. All right, now, this process is going to involve two steps, one of which is going to occur in the nucleus, and the other one is going to be out in the cytoplasm. All right, and so we are going to have to deal with RNA, and so let's just take a look at what it's made of. Now, RNAs, some RNAs are single-stranded, some are double-stranded. To be honest, most of the ones that we're going to deal with are single-stranded. All right, and they're also composed of nucleotides. Now, remember, what do the letters RNA stand for? What do they stand for? Ribonucleic acid. That's right. Okay? So, in this case, what is the sugar that you find in an RNA nucleotide? Ribose. Yes. And we also have phosphate again. And then we have the bases. And we have either purine or pyrimidine bases. Now, three of the bases are exactly the same as ones we saw in DNA. So, for example, we have adenine, cytosine, and guanine. They are back to visit with us again. But there's no thymine this time. Instead, it's uracil. So rather than having thymine, we substitute in a related base, a related pyrimidine base, uracil. All right? And uracil can pair with adenine, just like thymine does in DNA. All right? So just remember, you know, when it comes to the RNAs, there is no thymine. It's uracil instead. All right, so there we go. Now, there are several types of RNAs. Oh, and incidentally, this is just showing, here's thymine, like you find in DNA. Here's uracil, like it found in RNA. You notice they're very similar in structure. So both of them will pair with A. All right, now when it comes to RNAs, something must be in the way of mine. I think that card is kind of, yeah, I'll stick it here. Yeah, I can put it over here. All right, there, now it's out of the way. All right, there are types of RNAs that we're going to have to consider. One of them is what's called messenger RNA, and that is very typically abbreviated as little m RNA. It's single-stranded, and it is produced by a process called transcription. Transcription of DNA, in fact. And of course, where is that going to happen? It's going to happen in the nucleus. All right? So that messenger RNA gets made in the nucleus by a process called transcription. Now, the nucleotide sequence of the messenger RNA is complementary to the nucleotide sequence of the DNA of a gene. Want me to repeat that? I will. All right, nucleotide sequence of messenger RNA. Nucleotide sequence of messenger RNA is complementary to the DNA nucleotide sequence. To the DNA nucleotide sequence at the level of a gene. All right? So remember now, what was a gene? Sequence of DNA, right? We're going to make something complementary to it in RNA, and that's going to be a messenger RNA, as we'll see here in a bit. We'll do some examples. Now, once that messenger RNA is made, and it's made in the nucleus, it's going to take a little trip. It's going to go out into the cytoplasm. Now, how's it going to get from the nucleus to the cytoplasm? What does it have to go through to get out there? Well, remember there are some pores or channels, that nuclear membrane, right? It's going to have to go through one of those, all right? And once it gets to the cytoplasm, what it's going to do is link up with a ribosome. Now remember, what were ribosomes? They're organelles. What happens at a ribosome? That's where protein synthesis takes place. That's right. So that messenger RNA goes out into the cytoplasm. It links up with a ribosome. Now, we've got to align the ribosome with the messenger RNA. And there's another little RNA that helps us out with that, which isn't on this slide. I don't know if I can put it in this one or not. Yes, there it is. Ribosomal RNA. Now, ribosomal RNA is single-stranded. It's part of a ribosome. And it helps to align the ribosome on the messenger RNA. All right? That's what helps align the ribosome and the messenger RNA. And remember now, what were ribosomes made of? What kind of compounds were they made of? They have, obviously, ribosomal RNA. What else do they have? Protein. They're made of protein and ribosomal RNA. That's what they're made of. There were two subunits, right, to the ribosomes. What were they? What were the two subunits? What were their sedimentation constants? There was a bigger subunit called a 60S subunit, right, and a smaller called a 40S. And when you aligned them together and formed the single ribosome, what was the sedimentation constant for that? 80S, yes. Okay. And where did we find gobs and gobs and gobs of ribosomes in a eukaryotic cell? they were associated with another organelle that we had in there. Remember what it was? What was it? Rough endoplasmic reticulum. That's right, because that's where a lot of protein synthesis is going to take place. Okay, so we've had messenger RNAs. Those are made in the nucleus. They go out to the cytoplasm, link up with the ribosome. Ribosomal RNAs that are part of a ribosome that help align the messenger RNA with the ribosome. And then, if we go back, there's one more, and that's what's called transfer RNA. Now, what about that? Well, just as the name implies, it's going to shuttle something around. What it's shuttling around are amino acids. Now, there are 20 different amino acids. And there are going to be a number of different transfer RNAs that are specific for any given amino acid. Alright? And so, we're going to have transfer RNA picks up a molecule of its specific amino acid, and it is a specific combining. And it's going to shuttle that amino acid into the ribosome when we undergo the process of stringing amino acids together to form proteins. Alright? So these transfer RNAs, which are double-stranded, are going to link up with specific amino acids and actually carry or shuttle those amino acids into the ribosome, one at a time. But it goes very fast when it happens. Alright, now, the actual process of converting, if you will, a RNA nucleotide sequence into an amino acid sequence is called translation. And translation is going to occur at the ribosome. So we have first transcription, and transcription is the synthesis of a messenger RNA sequence that is complementary, has a nucleotide sequence complementary to the nucleotide or base sequence of the DNA at the level of a gene. Alright, so there you go. Now, let's just take a look at this simple little example first, and then we'll go with something a little more advanced. Alright, let's suppose we have a DNA base sequence of ATCG. Alright, there we go. Now, if we make a messenger RNA complementary to that, what will be the sequence of bases that we're going to find on it? Well, remember, adenine on DNA would pair with what on RNA? Uracil, U. Alright, thymine on the DNA would pair with what on the messenger? Adenine. Cytosine on the DNA would pair with a guanine on the messenger. And guanine on the DNA would give us a cytosine on the messenger RNA. And so, here we go. Uracil, adenine, guanine, cytosine. Complementary base pairing. Remember that RNA doesn't have thymine. So, if you've got an adenine on the DNA, that's going to specify uracil at that location on the RNA. Okay? And it doesn't matter which kind of RNA we're talking about. Those are the nucleotides, the bases that you're going to find. Okay? Now, that's transcription. That happens in the nucleus. Well, come on. Alright, now, once it's formed, as we said, the RNA, the messenger RNA, goes out into the cytoplasm. It links up with the ribosome. And the next step of the process takes place, which is called translation. And so translation is the synthesis then of a peptide or a protein based on the nucleotide sequence of the messenger RNA. Alright? So, DNA specifies messenger RNA, and messenger RNA specifies amino acid sequence. And when you string amino acids together by way of peptide bonds, what do you end up with? Well, you end up with either a peptide or a protein, depending on how many. What was the difference? Huh? What did you say? Size, yes. All right, what is the size difference? How many amino acids do you typically find if it's a protein? At least 100. Right, 100 or more. And if it's a peptide, it's typically 50 or less. You know, and in between, well, you can call it, I guess, a polypeptide. All right, so there you go. All right. And of course, as we said, this happens at the ribosome. Now, I trust everybody has handy their codon chart, which should look something like this. In fact, it ought to look very much like that. Okay, let me get mine out. And we'll have some fun with it. All right, now, here's the deal with this. On the messenger RNA, the messenger RNA is going to be read in a 3 base at a time sequence. And those 3 bases at a time are called codons. So codons are on the messenger RNA, and that's what you're reading off of this chart. Now, a codon is translated into an amino acid. All right? So, if we find, you know how this goes, like first letter, second letter, and third, all right? So, if we come up with something like AUG, which you'll see right here, and it's circled, that is labeled starch. Now, that is where the process begins. And the first amino acid that gets inserted is methionine. All right, they've written it out, and then they have the little 3-letter abbreviation there in parentheses. So MET is just simply the 3-letter abbreviation, standard abbreviation for methionine. All right? And you'll notice that each amino acid has numerous codons that specify. But the start is always that AUG. All right, so we'll look for it here in a bit. All right, now let's suppose that we have another codon, and let's say this codon is CAG. All right, find that one, CAG. What amino acid does that specify? Glutamine, right? Here it is. CAG, you find that 3-letter combination, and it's specifying a glutamine. Okay? All right, suppose we have GGG. All right? Here it is, and that would specify the amino acid glycine. Okay? Now, what if you come up with UAA? Well, UAA is right there. Look what's written beside it. Stop. There are 3 stop codons. All right? Now, what does that mean? Well, when one of those pops up during the translation process, the process stops. It terminates. That's the end of it. So if there are any codons coming after that, forget it. They're not going to get translated. They're going to be ignored. It starts as a start codon and proceeds until a stop codon shows up. And when a stop codon appears, that's it. That's it. The process is done. Okay? Now, shuttling in, of course, these amino acids are transfer RNAs. And they have a 3-base sequence on them called an anticodon. So codons are on the messenger RNA, and that's what you're looking at here on the chart. Anticodons are on the transfer RNA. And the anticodon is complementary to the codon. Okay? All right, so now, if we have as our start codon AUG, what would be the anticodon sequence on the transfer RNA that is shuttling in that methionine? Now, it's complementary to this, So if this is AUG, what would be complementary to AUG? UAC. That's right. Okay? Well, that's simple enough, isn't it? Heaven jazz. Well, let's have a little practice, shall we? Okay. So you hang on to your chart. And let's come back to this. And then when we get through all this step by step, I have a little animation to show you. It's so cakey, it's pathetic. All right, now here's the deal with our DNA. Now remember what we've got here. This is DNA. All right, it's a double-stranded molecule, and we'll say that this is part of a gene sequence, okay? Because a gene is just a part of that DNA that codes for a protein. Now, before we can go any further, we have to undergo transcription, right? To make complementary messenger RNA. All right, where's the transcription going to occur? It's going to occur in the nucleus. Now, two strands here. One of which is going to be what we'll call the coding strand. And that can also be called the sense strand. All right, coding strand or sense strand. Now, what about that? That's the one that serves as the pattern or the template to make the messenger RNA. All right? So the coding strand is the one that we're going to use to make the messenger. Now, the other one, the one that we have in green, is what we call our non-coding strand. And that one can also be called the anti-sense strand. And that one is not used to make a messenger. Okay? Not used to make the messenger RNA. Coding strand is used to make the messenger. That's the pattern to make the messenger. Anti-coding, non-sense, non-coding, no, isn't. What good is it? Why do we need it? When would you use it? When did we use that non-coding strand, I wonder? Well, it was used when we replicated DNA, wasn't it? Because remember, if we wanted to replicate that molecule of DNA, what would you do? Split them apart, come up with two newly synthesized strands to go with your two original parental strands, right? The semi-conservative synthesis of DNA. So the non-coding strand is used to replicate DNA, and it has another use too. It can also be used to repair the sense strand if it's ever damaged. Okay? Because the non-coding strand is not damaged, it remains intact, and the coding strand gets damaged, well, you've got the information there to put back what was originally there, right? You know, because it always does that complementary base pairing. So there you go. All right, so it's got a use. It's definitely got a use. But we're not going to use it now. So what are we going to do? We're ready now for transcription. We're ready to make a complementary messenger RNA. What are we going to do? We're going to have an enzyme come in and unzip these things, and I'm just going to take this non-coding strand, and I'm going to just put it aside, out of the way. And I got rid of it. All right, now, let's make a complementary messenger RNA. All right, so let's put our messenger RNA right over here. Oops. NA. All right, now, what nucleotides are we going to put together this time? All right, got an adenine there, so what goes over here? A uracil. And an adenine, and a cytosine, and a guanine, uracil, adenine, uracil, guanine. Right? There you go. The messenger is complementary to the DNA. Complementary. All right? And remember that on RNA, we don't have any thymine, so use uracil there to pair with adenine. All right? And there we go. And there's our messenger. Now, what happens with this messenger? Well, once the messenger is formed, it's going to go right out to the cytoplasm. And so we've undergone transcription here. So what's going to happen out here in the cytoplasm? What's the second step? Translation. All right. Well, let's start the translation process. So what am I going to do? I'm just going to rewrite that. So let's see, what have I got? U, A, C, G, G, U, A, U, G. All right. And I'll just stick it around this way. All right, there it is. Now there's the messenger RNA. It's now out there in the cytoplasm, and it's going to link up with a ribosome. Okay? All right, now, where does the process begin? It begins at a start codon. So, how are you going to recognize a start codon when you see it coming at you as broad as life and twice as natural? AUG. All right. Well, do we have an AUG? I think we do. We have down here. Well, that isn't going to do us any good. Well, tell you what, we'll fake it a bit. We'll pretend like it was somewhere up here. Let's take this 3 at a time. All right, now, UAC. All right, now, what amino acid would UAC translate into? Tyrosine. Okay, whenever I can find it, whenever I can get my bifocals on it. Oh, okay, there. Okay. Oh, yes, there it is. Okay. All right. All right, there it is. Now, next codon GGU, what would that give us? Glycine, okay. All right, then we have AAU. And that would give us asparagine. And there we go. And there will be others, but we don't know what they'll be, okay? Now, let's have some other fun here with this. All right, so over here, the transfer RNAs, let's put in their anticodons, shall we? Now, what do we know about the anticodons of the transfer RNA? They're complementary to the codon on the messenger, right? So if this one goes UAC, what's this one going to have been? AUG. All right? So that would have been the anticodon on that transfer RNA that brought in that tyrosine. Okay? All right, now the next one, the one that brought in the glycine, the messenger was GGU. What would be the anticodon sequence here? CCA. Yes. Okay? All right, and if the asparagine was the codon specified asparagine was AAU, what would have been the anticodon on the transfer RNA that shuttled that one? UUA. Yes. All right, there you go. All right? So the anticodons on the transfer, remember, are complementary to the codons on the messenger. Okay? All right. Now that's pretty simple, isn't it? All right, now here's the process again in a nutshell, remember. When we start in the nucleus with transcription. What is being produced during transcription? A messenger RNA, right? What serves as the template to make that messenger RNA? The DNA of the gene, all right? So that sequence of DNA as a gene gets transcribed into a messenger RNA. It's complementary to the nucleotide sequence of the coding or sense strand of the DNA. Okay? Once that messenger is made, where does it go? Out to the cytoplasm, links up with the ribosome, and then undergoes what's the next process called? Translation. All right? And translation just simply means translating or converting the base sequence of that messenger RNA into an amino acid sequence. Where does it begin? AUG, which is the start codon. Where does it end? Stop codon, right? And there are three possible ones. Now, this chart is not something to memorize. This is not to go in heart failure over. It will be provided. Okay? And I'm not going to say it'll be provided if you need it. I'm going to say it will be provided when you need it. Now, on the web page, I have put up a couple practice problems which I would heartily suggest you look at over the weekend and bring with you next week so we can take a look at them. And I've also gone ahead and put up the integumentary system outline, and when we get through that, we'll be ready for the next exam. So it's coming soon. And so, let us just take a look, and I don't particularly like this thing, so anyhow, I'm just going to snort this and go to my animation, which I think you will enjoy immensely. And to be honest, it just goes so simply once you take a look at all this stuff. So, let's see, bring that back. And I think I've got it ready to go, so let's cut the lights a bit. And there is sound with this. I think it'll play. I think the sound is on. So, let us see what we have. All right, now, I'm going to say another word or two here before we start. This is actually set up to work with prokaryotic cells, all right? And there are some slight differences. What are the big differences? Well, the ribosomes in prokaryotes are the same as what you find in mitochondria of eukaryotes. There's 70S ribosomes. But nonetheless, this works pretty much the same way. Methionine is the first amino acid inserted in a eukaryotic cell. It'll be formal methionine in prokaryotic cells. No big difference. Don't worry about that. The process, as I said, pretty much runs the same. And so what do we have here? This is the messenger RNA, all right, and that's one of the ribosomal subunits. And here's one of those double-stranded transfer RNAs getting ready to come in here. And of course, you can imagine where it's going to go, because there's the start codon. AUG and UAC is complementary to that, right? So that's how it's going to know where to go. So away we go.
In prokaryotic cells, translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formal methionine tRNA, and messenger RNA.
Now, remember, in a eukaryotic cell, what is the smaller subunit of the ribosome? What was its sedimentation constant? 40S. And the bigger one was 60S, so you just kind of have to make that substitution as we go along here. Okay.
The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA-carrying amino acids can bind. One is called the peptidyl, or P site, and the other is called the acceptor, or A site. There is also a third site called the exit, or E site, where transfer RNAs are released. The initiating transfer RNA carrying formylmethionine binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The formylmethionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of one codon, and the transfer RNA that carried the formylmethionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site, where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of one codon. As the shift occurs, the two amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
All right, now that's simple enough, is it not? All right, so now, where did the process begin? When a start codon was lined up with the P site of the ribosome and the transfer RNA carrying a methionine came in and entered that area, right? That was already there when the other part of the ribosome attached. All right, so where do you add new amino acids at the A site? All right, and then form peptide bond. That takes some energy, but you couldn't guess in your years what the energies were form the peptide bond. ATP, of course. And then the ribosome shifts a codon length down the messenger, and the empty transfer RNA goes out to pick up another molecule of the amino acid. You know, because these transfer RNAs get recycled, they're not wasted. So they'll go back. That first one will go right back, pick up another molecule of methionine. You can make the trip back many times. All right, and so then we're always adding at the A site new amino acids, and that continues until what shows up at the A site, a stop codon, and there the process terminates. All right, so it starts when you have a start codon at the P side of the ribosome. It ends when you have a stop codon at the A side of the ribosome. And anything that comes after that stop codon, ignored. Anything that comes before the start codon is ignored as well. Keep that in mind when you do your exercises. Anything that precedes the start codon is ignored. Anything that comes after the stop codon is ignored. All right? The only things that get translated are start until the one that precedes where they stop. And why does it stop? Well, there is no transfer RNA that's complementary to those stop codons. They don't have it, they can't be translated. There's no transfer RNA Now that's simple, isn't it? You know, wouldn't it be wonderful if everything was that easy? And you'll have a sheet to refer to. So, I mean, 90% of the effort's already there when you pull out the sheet. I mean, the merest modicum of thought is all that it takes. Now, pull those up and work on them and bring them with you to class on Tuesday. We'll take a look at them and then we'll get on to the scan. And it won't take long, so probably in another week we'll be about ready for another written exercise. Oh, you'll enjoy it immensely. All right, well, I think you've suffered enough for today.